Hi guys, welcome to another lecture on Medicinal Chemistry 2, and today we'll be talking about drugs affecting the renin angiotensin system and diuretics to a lesser extent. Alright, so this renin angiotensin system, or RAS for short, and hypertension, well, it is a hormonal system that plays a central role in the control of sodium excretion and body fluid volume. So we can therefore immediately see the connection between the system and hypertension as well as diuretics, right? So it interacts closely with the sympathetic nervous system as well as aldosterone secretion in the regulation of the blood pressure. So this is a system, starts off with renin release, and we have, conversion from polypeptides to smaller peptides, and peptides involved in the stimulation of the aldosterone secretion, which therefore leads to sodium and fluid retention and eventually blood pressure elevation. So this is a very well regulated and maintained system that occurs naturally within our system. So this is just a pictorial of the system that we just saw a while ago. Let's look at the legend for this. And so the blue arrow suggests secretion from an organ. So anytime we see a blue arrow, secretion. The this arrow with a plus in green represents a stimulatory organ, inhibitory organ, etc. So we need to understand this legend so that we can understand this diagram here. So angiotensin is secreted from the liver and renin from the kidney, and through a series of processes and enzymes catalyzes the conversion from angiotensin to angiotensin 1 and 2, and eventually to angiotensin 3. And we see in our previous diagram where both angiotensin 2 and 3 are involved in aldosterone secretion. So here, we have the adrenal gland cortex secreting aldosterone as a result of stimulation from angiotensin 2 and all of the effects that we just spoke about with regards to, let me go back to it, sodium and fluid retention happening, and we're just seeing a nicer image of this process happening. All right, so let's go into a little history. So in 1898, two scientists, Tigerstead and Bergman, demonstrated that when injected in a host, kidney extract produced a potent vasopressor response. The substance was named renin. So that's all the way back in 1898. Many years later, this substance was shown to require a cofactor to produce vasoconstriction, and then it was in 1939 where this hypertensive substance was isolated, identified as a decapeptide. So as you know, deca means 10 peptide, and later called angiotensin. This cofactor existed as an inactive precursor to angiotensinogen. So later, studies revealed that angiotensin existed in two forms: the biologically inactive decapeptide angiotensin 1 and the active octapeptide angiotensin 2. And as we saw later on, there was angiotensin 3 as well. So it's always good to go back in history to understand, where we're coming from in terms of all of this knowledge. So the precursor of angiotensin is angiotensinogen, and it's a glycoprotein of molecular weight 58,000 to 61,000, synthesized primarily in the liver and brought into the circulatory system. Renin is a protease that's clearly smaller molecular weight, whose primary source is the kidney, however. So angiotensinogen primarily from the liver and renin primarily from the kidney. And renin cleaves the leucine valine bond from the aspartic acid end of the angiotensinogen polypeptide molecule to release the decapeptide angiotensin 1. So the biochemical conversion continues with the cleavage of a dipeptide, histidine, and leucine. So this is basically what we're saying in a simpler diagram here. So renin causes this leucine valine to cleave from the acid end. So we have an amine end and we have an acid end. So from the acid end, angiotensinogen is cleaved, this leucine valine bond that produces angiotensin 1, which is a decapeptide. So we can count it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 amino acids, and that is basically what we just mentioned over here. So let's go back to this slide. So the biochemical conversion continues with the cleavage of histidine leucine, that is right here. So the first step is the cleavage from the acid end of angiotensinogen, and that produces angiotensin 1, which is further cleaved from the carboxyl terminal, so from the same acid terminal of angiotensin 1 by angiotensin converting enzyme. So from here henceforth, every time we mention ACE or we see ACE, we're referring to angiotensin converting enzyme, ACE, to form the octa, which is 8 peptide angiotensin 2, a potent vasoconstrictor. And angiotensin 3 is formed by removal of the N-terminal aspartate residue of angiotensin 2, a reaction that is catalyzed by glutamyl aminopeptidase. So in contrast to angiotensin 2, angiotensin 3 has a less potent but significant regulatory effect on sodium excretion by the renal tubules. So in our history, we mainly spoke about angiotensin 1 and 2. However, 3 is also potent, but not as potent as 2. So this primarily resulting, that is the angiotensin 3 being less potent but significant. This results in from the effect angiotensin 3 has in stimulating aldosterone secretion, a potent mineral corticoid. So let's go back to it here, where we saw that angiotensin 3 plays a role, a very important role as well. So even though it was the last to be discovered, it doesn't mean that it is not significant. All right, so that leads us to our first question, which is angiotensinogen is a, well, fill in the blanks. Angiotensin is a blank peptide. Angiotensin 2 is a blank peptide. So if you know the answer, let's see. All right, so angiotensinogen is a polypeptide, and angiotensin 2 is an octapeptide. All right, so let's look at for a second this model showing cleavage of the histidine phenylalanine residue of angiotensin 1 by ACE to form the octapeptide angiotensin 2 and the dipeptide residue of histidine and leucine. So we have a model here, right? And it's showing the important binding point at the active site of ACE, right? So ACE is an enzyme, which therefore means that it catalyzes a particular reaction. And the reaction that we're talking about here is the hydrolysis of this amide bond, or we might call it a peptide bond. It's an amide functional group. Anyway, so it's very important that we show or demonstrate the different binding points to show how it facilitates this hydrolysis. So the important binding points at the active site of ACE are a cationic site to attract a carboxylate ion. So here we have a positive cationic site on the ACE to attract a carboxylate. So this is a carboxylate ion. And a zinc ion that can polarize a carbonyl group of an amide function to make it more susceptible to hydrolysis. So we have a zinc here, right, that is now able to polarize, and this is our carbonyl functional group, and because it's positive, it will polarize the carbonyl group to make it more susceptible to the hydrolysis, and the hydrolysis therefore would cause eventually cleavage of this bond that I'm showing here. So once it cleaves, we end up with a carboxylic acid and an amine, right? So again, we're showing how ACE causes this cleavage to form, or histidine leucine dipeptide residue. So this is our histidine and leucine dipeptide residue because this bond has been cleaved. All right, so in the active site, there is a nucleophilic attack of the amide carbonyl by the gamma carbonyl group of a glutamic acid residue to cause hydrolysis of the peptide. So clearly a number of things has been happening in this ACE active site, right? So we already have this interaction here. We mentioned this interaction here. And the last thing is the nucleophilic attack on this carbonyl that would cause or further cause that hydrolysis that we mentioned earlier. So this is the glutamic acid residue that is found in the site. So anything that is in this particular region would be in the active site of the enzyme, and so this glutamic acid residue would attack the amide carbonyl, that's this one, by the gamma carbonyl group of the glutamic acid residue, and therefore cause hydrolysis of the peptide. All right, so the vasoconstrictive effects of angiotensin 2 are a result of many factors. So in the kidneys, it constricts glomerular arterioles, and the effects are greater on efferent arterioles than afferent ones. So afferent is the one going to, and efferent is the one coming out. So constriction of afferent, that's the one going in, results in increased arteriolar resistance, which raises systemic arterial blood pressure, right? So we're showing how everything is connected, and everything, we're showing all of the different factors that influences the vasoconstrictive effect of angiotensin 2. All right, so angiotensin 2, as we saw earlier, plays a primary role in regulating aldosterone secretion. Upon stimulation by the renin angiotensin system, which we can call RAS, aldosterone is secreted by the adrenal cortex, which we also mentioned, and elicits its effects at various sites, including the nephrons. So it is responsible for the reabsorption of sodium into the bloodstream, and therefore this results in increased levels of sodium in the plasma, which in turn result in increased blood volume and vascular resistance. So if aldosterone levels in the body become too high, then symptoms such as elevated blood pressure or heart failure can occur. So this is something that our bodies have to definitely regulate. So in addition to that, the regulatory action of RAS in controlling sodium and potassium balance and arterial blood pressure is modified by vasodilators called kinins, right? And these are proteolytic enzymes that circulate in the plasma that form kinins. So kallikrein, which is, if you look at this diagram here, kallikrein is here, is activated in plasma by noxious influences to act on a kinin, this kinin called kallidin. And if you do read widely, you will see that there are a variety of spellings for these two words, which is, so kallikrein is activated in the plasma to act on kallidin, which is converted to bradykinin by tissue enzymes. So bradykinin, right, so that's converted from kallidin through kallikrein. So bradykinin enhances release of the prostaglandins, right? So PGE2 or PGI2 within certain tissues to produce a vasodilatory effect. So we can see therefore that our body is so regulated so that we have a system in place that can cause vasoconstriction, and we also have a system in place that can cause vasodilation. So bradykinin is converted to inactive products by ACE, that's here, and other carboxypeptidases. So although ACE causes activation of angiotensin and inactivation of bradykinin, actions that appear to be opposite the balance of the system seems to favor vasoconstriction. All right, so let's go on to another question, which is show the hydrolysis of histidine and phenylalanine. And it'd be nice if you actually know these two amino acids and join them together so that you can show the hydrolysis. Well, if you just chose to use R to represent all of the other functional groups, that would have been fine. But the key point is here that we have an amide functional group, NHCO, C double bond O, and when it undergoes hydrolysis, we have hydrogen that's added to the nitrogen, and we have a hydroxyl that is added to the carbonyl portion, and so we end up with an amine and a carboxylic acid, as we saw in the ACE active site. All right, so because the renin angiotensin system plays such an important role in 1, regulating kidney function, 2, aldosterone release, 3, electrolyte balance, 4, blood volume, it is easy to recognize why targeting this pathway is beneficial in the management of high blood pressure and heart failure. In other words, since we have a general understanding of the system, then we can therefore understand that if we make drugs that target particular aspects of the system, then we can therefore play a role in regulating or blood pressure or other illnesses. So let's look at categories of drugs affecting the biosynthesis or actions of angiotensin 2. So the first target that we're going to look at is angiotensin 2. As we saw in our introduction, it is one of those parts of the system that plays a crucial role. So if a drug can impact it, then it would have some amount of success. So at present, there are 3 main categories of drugs affecting the biosynthesis or actions of angiotensin 2. They are the renin inhibitors, the ACE inhibitors, and the angiotensin 2 receptor antagonists. All right, so we're going to be looking at a few types of drugs. The first type that we're going to look at is angiotensin-converting enzyme inhibitors. So these are our ACE inhibitors. So we have some examples here. We have Captopril, Lisinopril, and Enalapril, etc. So there are a couple other prils in the series, and we have some images here of Captopril, the label, and packaging for the other 2. So again, we try to look at where it started, and then hopefully we can get a better appreciation for the process of discovery. So towards an ACE inhibitor, although the enzyme ACE could not be isolated, the design of ACE inhibitors was helped by studying the structure and mechanism of another zinc metalloproteinase an enzyme called carboxypeptidase, right? So in other words, or repeating what I just said, they could not isolate ACE, right? So they decided to employ a similar enzyme that was related to see if they could try to mimic a similar process or the impact that would perhaps be the same. And as we saw with our active site, we saw that zinc was a necessary component in our active site. And so they found a similar enzyme, a zinc type enzyme, and they tested it to see or what could impact that active site. So this enzyme splits the terminal amino acid from a peptide chain and is inhibited by I-benzyl succinic acid or L-benzyl succinic acid. So this is a structure of L-benzyl succinic acid. So this is succinic acid and this is the benzyl portion here. So this is a succinic acid, this is benzyl portion here. So this enzyme, as they said, splits the terminal amino acid. That is, here we have amino acid 1, amino acid 2, amino acid 3, and it splits the terminal one from the acid end, and the enzyme is inhibited. Let me just read. This enzyme splits the terminal amino acid from the peptide chain and is inhibited by L-benzyl succinic acid. And as I said, that's the structure of L-benzyl succinic acid. So the rational development of Captopril as an inhibitor of ACE was based on the hypothesis that ACE and carboxypeptidase A functioned by similar mechanisms. And so it was noted that D2-benzyl succinic acid was a potent inhibitor of carboxylase A, but not ACE, right? So even though they tried, that was the basis for the development of Captopril. So they basically modeled, so let's just start over so that we can have a clearer picture. They couldn't isolate ACE, right? Because that would be the ideal thing to find a target, right? But they couldn't isolate it. So they found a similar type enzyme that does something similar to ACE, right? And they found that this molecule inhibited it. And so they decided to model, to make a similar type compound, which is Captopril. So it was noted that the 2-benzyl, or this compound here, was a potent inhibitor of carboxypeptidase A, but not ACE. So that's an interesting find, that even though they modeled it after that, it wasn't able to inhibit ACE. So that is the genesis of one of our angiotensin-converting enzyme inhibitors, Captopril. So I found that interesting, and I hope that you do as well. So, however, as ACE splits a dipeptide unit from the peptide chain rather than a single amino acid, right? Because we know this already from our introduction. These groups are likely to be further apart, and so an analogous inhibitor to benzyl succinic acid would be a succinyl substituted amino acid. So the next step was to choose which amino acid to use. So unlike carboxypeptidase, ACE shows no specificity for peptide substrates containing any particular C-terminal amino acid, and so the binding pocket for the C-terminal side chain must be different for the two enzymes. So we kind of mentioned this already. The two enzymes were different. However, it's still significant, it's still noteworthy because one thing essentially led to another that was effective. So another question is, what are the functional groups of Captopril? So this is the structure of Captopril, and you should be able to identify all functional groups. All right, so for your assistance, I included a little resource with the main players of organic in terms of organic functional groups. So obviously there are others, but these are the main ones. So if you didn't say thiol, right? If you didn't say amide, if you didn't say carboxylic acid, if you didn't say alkane, then you might want to check out this box here. All right, so Captopril. So Captopril, it has a carboxyl group. As we saw, we just identified functional groups. On a proline, and a thiol group was introduced to enhance the binding to the zinc ion of ACE. So this again is the active site of ACE, and they're showing the interaction of Captopril with the different binding points. So the important binding points at the active site of ACE are thought to be an arginine residue, that's down here, which provides a cationic site, and of course you must know cationic means plus, that attract plus or positive, that attracts a carboxylate ion here, and a zinc ion here, which can polarize a carbonyl group of an amide function to make it more susceptible to hydrolysis, as we have seen further in our introduction. And of course there are some hydrophobic pockets which lie between these groups in the active site, as does a functional group that forms a hydrogen bond with an amide carbonyl, right? So even though we're not seeing it, there must be some hydrogen bonding between our amide carbonyl here and our active site. So guys, this is an oversimplified diagram of our active acid. You must understand that it's much more complex than this, but we're just showing what they think are the important binding points of Captopril. So this is medicinal chemistry. So as usual, if we can put in a little synthesis, we try to. So the synthesis of Captopril, well, there are several methods of synthesizing Captopril. One method, which I have on screen here, is to take the T-butyl ester of proline, and it is appropriately labeled, and react it with the carboxylic acid in the presence of a coupling agent DCC to form amide 2. All right, so this is our carboxylic acid, and this is our T-butyl ester, and in the presence of our coupling constant, we basically have a new bond between our nitrogen and our carbonyl here. So what is missing, and this is how you're supposed to be looking at this, is actually a water molecule. So there's no longer a hydrogen, there's no longer a hydroxyl. There's just a new bond here between the carbonyl carbon and the nitrogen. Once this is formed, the T-butyl and acetyl protecting groups can then be removed in the presence of acid and base respectively to give Captopril. So essentially, these are referred to as protective groups. Protective groups are groups that, if they are not there, there would be reactions involving, the sulfur or the acid. So chemists try to put on different groups, as we see here, that we call protective groups, that we can carry out a particular reaction and then remove them so that we can get our desired product. So again, our protecting groups are T-butyl and acetyl or acetyl. The acetyl functional group is here, and it's protecting the sulfur, and the T-butyl is here, and it's protecting the carboxylic acid, right? Because it would get involved in the reaction, which is not desired, right? So they put on these two groups so that we can carry out the coupling reaction, and then after that, they can use acid to get rid of the tert-butyl, and then base to get rid of the acetyl, and we end up with our Captopril. Right, so two important points here. The protecting groups used, we can put them on, we can take them off to protect a particular functional group. And of course, our bond formation, our new bond formation between our nitrogen and our carbonyl carbon here, and the loss of water and our coupling constant caused the entire synthesis. All right, so another drug that we are mentioning is Lisinopril. So Lisinopril, and we have the actual name of it, like the chemical name of it, is a lysine derivative of enalapril, the active metabolite of enalapril. Like all ACE inhibitors, it is an active site-directed inhibitor of the enzyme, with the zinc ion used in an effective binding interaction at a stoichiometric ratio of 1:1. The pharmacological effects of lisinopril are similar to those of captopril and enalapril. So here we see the structure of lisinopril, and similar in structure and enalapril. They both have an ester, the same ester group here, and they both have this aromatic portion and or amide portion as well, and our carboxylic acid. So we can readily identify similarities and differences between our captopril or lisinopril and or enalapril. And well, of course, we have our lysine, because we said is the lisinopril is a lysine derivative of enalapril, right? So here we have, at this position, we have the lysine being present here. All right, so many new ACE inhibitors became available for the treatment of hypertension following the clinical effectiveness of enalapril. And on this slide, we have several of these ACE inhibitor prodrugs. And at this point in your medicinal chemistry journey, I'm sure you know the definition of a prodrug. Enalapril is a non-thiol containing ACE inhibitor devoid of the side effects of rash and loss of the sense of taste characteristic of the thiol containing compound Captopril. All right, so we're going back to the structure here, and perhaps we should go all the way back to Captopril, where we saw that it has a thiol portion here. So enalapril is a non-thiol containing, as we saw, ACE inhibitor devoid of the side effects of rash and loss of taste or sense of taste characteristic of the thiol containing compound Captopril. So with the exception of phosphorus containing Lisinopril, which the structure is here, these antihypertensive agents have a 2S-aminophenyl butyric acid ethyl ester moiety differing only in the substituents on the amino group. All right, so clearly we have our ethyl ester moiety. So we have, let me just try it out. So, or, because we're not seeing it very clearly, we have our CH2, CH3, and that is our C2H5. So we have an oxygen that's bonded to an ethyl, so all of them have, that portion there, or ethyl ester moiety. So we have to note that part, and they all have, for the most part, the aminophenyl butyric acid portion. So this is our phenyl portion, and we have the aminobutyric acid, so that's generally 4 carbons moiety. Right, so clearly they all are similar in structure, so not the same, but they have these similar components. But we just need to have an idea that so we don't have to know the structures, but we need to know, like, for example, the major functional groups, and like, for example, what we said here, that most of them have the aminophenyl butyric acid ethyl ester moiety, and they vary on the substituents on the amino group. All right, so continuing here with our ACE inhibitor prodrugs, we have other structures here. So we have benazepril, and I guess the point of this is to show that all of them has some type of pril in the name, and all of them have some type of similarity in structure. All right, so they have the common property of acting as prodrugs because they are converted to the active enzyme inhibitor following absorption and metabolism by liver and intestinal enzymes. So these drugs, like the prototypical drug Captopril, are used in the treatment of mild to moderate hypertension, either alone or in conjunction with diuretics or calcium channel blockers. So here we have a list of, I think, yeah, we had all of these structures on the slide previously. So we have there the prodrug. So what we have been seeing is the prodrug. All of the structures that we have seen are the prodrugs, and the metabolite is the prelat of all of them. So all of them end with pril. And the metabolite, which is the active form of the drug, is the lat, prelat. And we have the mode of excretion, which is mostly renal, and metabolite plasma half-life in terms of hours, roughly from 10 to 13. Enalapril having much less than the others, and or metabolite protein binding, pretty high percentage for the most part. All right, so Enalapril Maleate, and this is the chemical name of it and the brand name. It's a long-acting ACE inhibitor. It requires activation by hydrolysis, because as we said, they're all prodrugs, and they all require activation by hydrolysis of its ethyl ester. So we did show the ethyl ester on the groups to form the diacid enalaprilat, as we just saw in the table. So here we have enalapril, and we have our ethyl ester here. And as we saw, or as we can see in the product, or enalaprilat, there is hydrolysis. So we have the carboxylic acid, which is the dicarboxylic acid. So there was one already, and now there is two. So let me just repeat. So it requires activation by hydrolysis of its ethyl ester to form the diacid enalaprilat. Enalapril is devoid of the side effects of rash and loss of taste seen with Captopril. The absence of the thiol group in Enalapril Maleate may free it from these side effects. The half-life is 11 hours. So again, because it doesn't have sulfur, it doesn't have the rash and loss of taste side effect. So that would have been an awesome improvement. All right, so moving on to our angiotensin antagonists. So there are 4 different subtypes of angiotensin 2 receptor subclasses identified to date, and that includes 81 to 4. These belong to the class of G protein coupled receptors and are responsible for the signal transduction of the hormone. Studies have demonstrated that stimulation of AT1 receptors by angiotensin 2 results in 1. Vasoconstriction, 2. Aldosterone synthesis and secretion, 3. Increased vasopressin secretion, 4. Decreased renal blood flow, 5. Renal tubular sodium reuptake, and several other physiological events. So the administration of competitive antagonists that inhibits angiotensin 2 at the AT1 receptor will produce a vasodilator effect, because the substrate for this receptor is an octapeptide. Much of the earlier work was performed by using various peptide systems. One such agent that was used was sarolacin, and it's also an octapeptide that differs from angiotensin 2 by 2 amino acids. So I don't know if you guys remember or recall the 8 amino acids, but it would be nice if you could look at this now and say, okay, that is there, that was there, etc. Right? So definitely it differs by 2 amino acids. So this agent's use was limited because it had some partial agonistic properties. right? So that clearly would have been an issue. Nevertheless, it served as a lead in the development of other agents that are useful at antagonizing the angiotensin 2 receptor, right? And as we said, when we looked at the Captopril model, that even though, what we hoped would have been effective was not effective, one thing led to another thing that was effective. And I suppose it's a similar situation here where they use this octopeptide that turned out to be partially agonistic when the intention was to find an antagonist. All right, so we have another question. Identify cerulacin and angiotensin 2. So we have the list of both our octopeptides, and we have the list and of both of them there. Can you identify which is cerulacin and which is angiotensin 2? Well, if you had the one that started, well, if you had it as such, A representing angiotensin 2 and S representing cerulacin, then you would have been correct. I also took the opportunity to get you familiarized with your amino acid. So at least try to know the ones that we are dealing with right now: ASP, ARG, VAL, TYR, ILE, HIS, PRO, PHE. Those are the amino acids found in angiotensin 2. So at least try to familiarize yourself with them, because we have mentioned them. We mentioned glutamate in the receptor, aspartame, like we mentioned them. So at least try to familiarize yourself with not just the 3-letter abbreviation, but the actual name. All right, so the most significant lead in the development of this class came from a series of imidazole 5-acetic acid derivatives that attenuated pressor response to angiotensin 2 in test animals. The molecular modeling revealed that the imidazole 5-acetic acid could be exploited to mimic more closely the pharmacophore of angiotensin 2. The first successful agent to be developed through this method is losartan, and the structure is seen there. These tend to be biphenyl methyl derivatives that possess certain acidic moieties, and we have the blue circle there containing the acidic moiety, which can interact with various positions on the receptor, much like the substrate angiotensin 2, right? So here we have our imidazole 5-acetic acid that can play a part, as it can be a angiotensin antagonist. So as we mentioned, losartan, and that is the brand name and the chemical name. It was the first non-peptide imidazole to be introduced as an orally active angiotensin 2 antagonist with high specificity for AT1. When administered to patients, it undergoes extensive first-pass metabolism, with the 5-methanol being oxidized to a carboxylic acid. So that is the 5-methanol. being oxidized to a carboxylic acid. So in the metabolism, this CH2 is now a carboxylic acid. This metabolism is mediated by CYP2C9 and 3A4 isozymes. The 5-methanol metabolite is approximately 15 times more potent than the parent hydroxyl compound. That is significant. So that metabolite, so we wouldn't call losartan a prodrug because it is active, right? And it's just that the metabolite of it is more potent than the parent hydroxyl compound. So because the parent hydroxyl compound has affinity for AT1 receptor, strictly speaking, it is not. Oh, so that's kind of what I said, right? So the parent compound is active, so we cannot call it a prodrug. So angiotensin antagonists, we have several variations here, and this is really, again, not for you to try to remember the structures, but to remember to look at the similarities, the biphenyl portion that we mentioned, if it has a methanol portion, etc. So compare and contrast the structures. All right, so another class of compounds are the renin inhibitors. So renin is a circulating enzyme that is secreted by the kidneys in response to decreases in glomerular filtration rate, which is typically a result of low blood volume. So it is a 340 amino acid protein that participates in the RAS. Inhibitors of renin have usefulness in managing cardiovascular diseases such as hypertension and heart failure because of their ability in reducing the influence on the renin angiotensin system through reduced biosynthesis of angiotensin 2 or other bioactive intermediates. Because based on our introduction, which if you need to rewatch, you probably need to go back there to see how renin impacts the whole RAS system. So if you inhibit renin, then you literally inhibit the other hormones in the process. So although this drug target has been investigated for more than 30 years, it represents the first new class of orally active antihypertensive agents and joins at least 7 other classes of antihypertensive drugs. The other classes include diuretics. So we have an example here of a renin inhibitor, which is Aldosterone, and as usual we have our trade name in brackets, Tecturna, and we have our chemical name here, which is clearly very lengthy. So Aldosterone is the first renin inhibitor introduced into the US market. It was approved by the FDA in 2007 for the management of hypertension. The trade name for Aldosterone in the UK is Racilic. So you know that the trade name will vary from locality to locality. Oh, well, we didn't look at the structure. So looking at the structure now, what do you notice? What are the functional groups? And is there any similarity or differences to renin itself? These are the questions that you should be asking. What makes it structurally, what makes it an inhibitor of renin? All right, so the final group that we're going to look at is aldosterone antagonists. They are, as the name implies, receptor antagonists at the mineralocorticoid receptor And antagonism of these receptors inhibits sodium resorption in the distal tubule of the nephron, which in turn interferes with sodium-potassium exchange, reducing urinary potassium excretion. So overall, this results in a weak diuretic effect and decreases cardiac workload. So agents that block aldosterone may be used in the management of hypertension or heart failure. The two agents approved for the use in the United States are spironolactone and you have to help me with this one, eplerenone or Inspra. So we have the structure of aldosterone here, and we're going to see it again on this slide. So we mentioned already the two agents approved in the US. So these compounds, and well, here we have the structure of the two compounds approved in the US, and we have the structure of aldosterone here. So what you want to look at, guys, is similarities and differences in terms of structure. So these compounds have an A-ring enone, like aldosterone. So I have a generic structure of a steroid here, and it's labeled ABC. So the numbering is there and the different rings, A ring, B ring, C ring, and D ring. So on the A ring, we have an enone. So an enone is a double bond and a none. So a ketone and an alkene. So ene for the alkene and one for the ketone. So all of them have the ring A enone, or both of them, I should say. Essential for recognition by the receptor. But the 7-alpha substituent and the D-ring spirolactone provides structural elements that lead to antagonism. So our 7-alpha substituent, so clearly in aldosterone there is no 7-alpha substituent. Here we have a 7-alpha substituent, and we also have a 7-alpha substituent here. And the D-ring, so this is the D-ring here. This is a spirolactone. Both of them, so these two, this is how both of them differ, or the main way that they differ in structure from aldosterone, right? So they have something similar to aldosterone, which is the ring A enone, and they have something different from aldosterone, which is a 7-alpha substituent and the D-ring spirolactone. So one makes it one, the enone causes it to be recognized by the receptor, and the differences lead to the antagonism. All right, so finally, guys, the spironolactone is an aldosterone antagonist of great medical importance because of its diuretic activity. And just to mention that a diuretic is any substance that promotes diuresis, that is the increased production of urine. So this includes forced diuresis, and there are several categories of diuretics. All diuretics increase the excretion of water from bodies, although each class does so in a distinct way. So this is the same image of the compound that we had on the previous slide. The only addition here is a snippet from our introduction where we saw that aldosterone secretion causes sodium and fluid retention. So clearly, if you antagonize it, it will cause the opposite of that, which is diuresis. And of course, other effects. All right, so this is where we conclude our chat, and I just want to leave you with one question, which says the renin angiotensin system plays an important role in regulation of blood pressure. For that reason, this pathway encompasses the conversion of an approximately 400 amino acid sequence to a decapeptide, then to a biologically active octapeptide, and is an excellent target for drug therapy to manage hypertension. Describe the starting protein and its conversion to the biologically active metabolite. Be sure to include the enzymes involved in the conversion of the protein and peptides. So come prepared to answer this question. All right, so thank you guys for watching and see you next time.